Hi everybody, welcome to our third podcast in our information domain. This one's going to focus on biotechnology. Just like the previous one, we're going to break this up into 3 sections, so feel free to break it up yourself. Feel free to sit down and watch the whole thing. You will see the whisks below have been broken up into 3 parts. So here we go. And I will say though, in terms of the parts, they are not evenly dispersed. We've got part 1 and 2 are probably bigger, and part 3 is a little bit smaller. So just to kind of give you guys a heads up on that. And just like before too, I will point out particular videos that I think you guys will probably want to go watch. You can do that by going back in, opening up the presentation and going directly to them, okay, because they're linked right in there. So without further ado, we are going to look at biotechnology. Without a doubt, probably some of my favorite stuff, and we're going to do some cool stuff. So let's jump right in. So here are the big things we need to be able to answer and understand. So the relationship between science and technology, how developments in genetics led to these new technologies, the implications of these to society, and kind of the big thing that can create some interesting discussion is what are our ethical and moral responsibilities as scientists with regard to this, to kind of quote from Jurassic Park, just because, we can, should we, and that's kind of the big thing that kind of comes out of that. So let's go. So the first part, in part 1 here, we're going to look at the tools and techniques used in common biotechnology and kind of explore. This is a little survey of some of them. We're going to do a couple of things, what it is, how they work, and what they are. Or I guess to reverse that, what it is and how it works. So the 2 big questions we're gonna deal with. So we're gonna start with restriction enzymes. These are used so often and in multiple types of biotechnology tools. So what are restriction enzymes? They are enzymes that work to recognize very specific sequences in the DNA strand, particularly what we call palindromic sequences. And these enzymes are naturally occurring in bacteria. They're a defense mechanism to snip up DNA that shouldn't be there. So we have exploited them by removing them, and we can use them to cut up DNA. And so they cut, and then they create what we call the sticky end, because when they cut at that palindromic site, they always make kind of this zigzag cut through the double helix, as you can see here on the bottom. And what you're seeing here is probably one of the most well-known restriction enzymes called EcoR1, and it has a very distinctive GAATTC palindromic site. So again, palindrome, forwards, backwards, and it cuts between the G and the A on the 2 sides of the DNA strand, leaving you with open TTAAs on either side, as you can see. So that's what they is. Okay. Officially, we refer to them as restriction endonucleases. They cut at very specific sites, and in essence are that idea of that bacterial immune system. So here it is in sort of a space-filling model of EcoR1 wrapping around the DNA molecule, kind of reaches right in and goes and cuts it, which is kind of cool. One of the things that I will say is all of the restriction enzymes that we have found and isolated are named after the bacteria that they come from, as well as the number in sequence that they were found. So EcoR1 is the first restriction enzyme found in E. coli. So the beauty of them as an enzyme, as we know that they're so specific, is that each restriction enzyme cuts at a very specific restriction site. So that palindrome, not any restriction enzyme works on any palindrome. They're very, very specific, and any time that palindrome comes up, so anytime GAATTC shows up on a DNA strand in the sequence, if EcoR1's there, it's going to cut it. So you could have it show up 4 times on a DNA strand, you're going to have 4 cuts. Pretty easy how it works. This was the winner of the Nobel Prize in 1972 for its discovery, and so you can what's cool is I can use EcoR1 to cut my DNA or to cut whoever's DNA, and they are universal in that case. So from different sources we can use it, and then the cool part of this is we can use it for inserting genes, because if I cut my DNA let's say with echo R1, I'm going to be left with my sticky end, that TTAA sticky end, or AATT. If I cut a gene out from another organism, those sticky ends are going to have the same sticky ends, and I can insert it in and then use ligase, okay, to help insert and be able to have a combination. So I can basically recombine. So genetic recombination stems from this ability to use restriction enzymes to do this. Again, those of you that had experienced piglo before, the plasmid we used to insert into the bacteria had been a synthesized plasmid having been chopped up and with that piglo gene inserted into it. And this is how we create human humulin, which is the synthetic form of insulin created by bacteria using our gene. And, so we splice it in and we create it and then we put it in and then the bacteria start pumping out that. So it's pretty awesome. Okay. There's another type of biotechnology tool called vectors. They are bacteria with the plasmids, and we use these vectors. These molecules can be modified to replicate in other sequences. So we can use bacterial plasmids, phages, viruses, artificial chromosomes, and they're super useful for plasmids because you can have an origin of replication, you can have multiple cloning sites, the gene that can enable the screening and successfully taken on. So again, things like we can insert and create plasmids to be able to do this. So again, that idea of creating different types of plasmids that have particular things, we can insert genes wherever we want. Okay, so this is what makes them extraordinarily useful tools. They have to have these kinds of things, and you can see this is actually a plasmid here that we would insert for things like ampicillin resistance, like in the piglo plasmid we used. So that allows us to have particular screening mechanisms. If we have an ampicillin resistant gene on the plasmid, and I insert it into a bacterial cell, and those bacteria cells can grow in the presence of ampicillin, I know that they took the gene up because they're resistant. Makes it pretty easy, right? So there's size limits though. Plasmids are tiny. They can't be really big at all. So if you're going to get more genes, you have to create artificial chromosomes. So literally synthesize piece by piece by piece by piece the DNA code, right? And here's what was really cool. In 2010, Craig Ventnor, his institute, he was one of the private funders for the Human Genome Project back a while ago, and his institute created a cell that had a completely synthesized genome. Yeah, okay, so fully synthesized, meaning man-made created genome that they inserted and created a cell, and it divided. That was the thing. It wasn't so much that they could put it into the cell, it was the fact that it took it on, turned the information on, and the cells divided. Right? Crazy. There's also techniques we use to read and to be able to look at and see. So like if I use restriction enzymes, how do I know they work? If I use vectors, how am I going to know they're going to work? So there are some of the tools that we use called electrophoresis. We've all, I think, experienced electrophoresis. It's a mechanism of a way to separate fragments of DNA. You can also use it for proteins too, but it's got a little bit of a different reason behind why it works. But for DNA, it works because I can run an electric current through it, and DNA being negatively charged will attract towards the positive terminal end if I run electricity through it. And based on size of those pieces of DNA, they're going to travel at different speeds through that gel, pulling them apart from one another. And that's how it works, which is really, really cool. And so you put it in, you place it into a gel matrix, usually agarose. It migrates towards the positive end. Smaller pieces move quicker, bigger pieces move slower, and what you end up with is what's known as a DNA fingerprint, just like right here. So that pattern that develops can be unique depending on what you use. Now, this gorgeous hot pink, for the bands is due to a staining mechanism. Once you stain those fragments of DNA, each one representing okay, just so to be clear, each band is not a single fragment. It is many of that size. The darker the band, the more you have. Okay, so here though we're using ethidium bromide, and I would love to use ethidium bromide. Problem is it's really nasty and it's cancer causing, and we can't use it in here. But what you do with it, once you stain it binds to DNA so well, and then you put it under UV light, and pow, hot pink bands show up. So I've used it; it's pretty cool. So we will be doing electrophoresis as well, and so labeling, okay. This is how we want to identify particular sequences of interest. So that's what we do when we start dealing with labeling. Okay, when I want to find out what these actually mean, maybe I use it in forensics, or if I'm trying to find out where samples came from. Okay, so fragments that are isolated, get denatured, strands separated. They're exposed to a complementary oligonucleotide probe that you can tag with radioactivity, right? And then that bonds to complementary sequences along the fragments. So kind of let's look here, right? So here's the technique of it. DNA and the restriction enzyme, right? Boom, boom, boom. You get your fragments, you run your fragments through the gel. There's my gel, okay? So now I take the gel, right, and I use all this solution, and I basically really, really, really flatten it out and blot it onto paper, onto radioactively labeled paper, right? The radioactivity, okay, the labeled probe for the beta globin, okay, you can put it on this, create this blot, label it, put the film over it, and then you can have, a thing to look at. So yeah, that's kind of part of what you can do. In the bottom half of the technique, you use all of these sort of multi-weight probes, you put it in, and that's where you get it. So you get the probe detection from there. Not one we're going to focus a ton on, I guess wanted to really just introduce you to it. So that's labeling. Okay, PCR. We will get to do this too, polymerase chain reaction. This is an awesome method. The winner of the Nobel Prize in 1993 for PCR, and what it is a big old DNA copy machine. Okay, ways to produce multitudes of DNA fragments in a very short amount of time, and when you have only small, minute quantities, in theory, you just need one, and you can copy it. So without the use of cells. So as long as I have the DNA, I don't need cells to do it. Okay, so very cool. So this is, we say thank you, Carrie Mullins. The thermocycler is what you need. I like ours better than this picture because it's purple. What can I say? So essentially it's a very fancy oven. It raises temperatures. It goes up way up high, brings it low, brings it back up again, and it can do multitudes of cycles based on however long you want it and whatever you want. So specific temperatures and very, very, very specific ways and for how long. That's all a thermocycler is, okay? And so it's just fancy for doing that. It's what you put in is what makes, I guess, the magic happen. So we use an enzyme because we know DNA polymerase is necessary for attaching free nucleotides to template strands of DNA. However, in order to make this work without cells, to make the DNA come apart, I actually have to heat it wicked high. Now, if I heat it wicked high using our enzymes, they're going to denature. That's going to be a bummer. So, because at high heat, breaks the hydrogen bonds and pulls the helix apart. So if I do that, yeah, I'm not going to have any enzymes to do what I need it to do. In comes Taq polymerase. Taq polymerase is isolated from a bacteria that is found in the hot springs of Yellowstone National Park, meaning it can exist at crazy hot, high temperatures. So we pull out its polymerase and we use it because it's stable and can work at the temperatures necessary for PCR to function. Love it. So you get your target DNA sample that you want, okay? You put everything all into your tube, so you're putting in primers that are going to bracket the sequence because you're not going to copy the entire genome. That would be silly. So you're going for the sequence you want. You attach these primers that bracket the sequence that you want on either side, okay? Then those primers, right? That's the docking mechanism for the polymerase, comes in and will attach. So along with the primers, you're going to put in the polymerase, you're going to put in all your free nucleotides, shake in all your A's, your T's, your G's, and your C's, get some coenzymes in there and everything, mix it all up into the thermocycler. Okay, so it programs and it can run, and in one sequence overnight, you can make as many as 2 to the 30th, okay, to the 30th copies of that DNA overnight. Okay, huge amounts just by raising and lowering and raising and lowering, and that exponential grows, right on up it goes. Okay, so here it is kind of in terms of what happens. Okay, let's go there. So the first part is I want my genomic DNA. There's a sequence I want, okay? And so what we do first is you have to denature the DNA. Now here's the thing that's different about denaturing of DNA than protein. When I denature DNA, all that means is I'm breaking the hydrogen bonds between the two strands. So I do this, pull it apart, right? When it cools, it goes back together again, thankfully. So you can pull it apart, but if you cool it back down, it sweeps right back up again. So in the denaturation, so this is one single cycle of PCR. You denature, and this is between high 90s degrees Celsius. Separate it. Then you have what's called the annealing step. We drop the temperature down because if you don't, then the primers don't attach. So we go back down to about 50, 65 degrees Celsius, get the primers to attach to the sides that you want, so they attach. Then you get your extension. Bring it back up a little bit to a happy optimal temperature for Taq polymerase. That attaches and it begins to replicate your target sequence with the free nucleotides floating around in the strand. Okay, so once you have that, boom, 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 every time, every single cycle you're going to do that. So if I start with 1, we know how replication works. I get 2, those 2, I get 4, those 4, I get 8, 16, keep it going, okay? That would be one cycle. So the denaturation, the annealing, the extension, okay? Guess what happens next? We bring it back up to 90, those pieces separate, we go to the annealing, so that thermocycler is constantly raising and lowering temperatures. I love PCR, I can't wait to do it. So here is where you might want to just make a star where you would want to go back into and watch a couple of videos. Some are fun. I'm not going to lie, I had a little fun. And where you can watch some fun stuff going on with PCR. Okay. So other things that we can do is create gene libraries, and these are collections of DNA sequences that get stored in vectors. Again, stored in a plasmid, stored in, however we're storing. That's what the vector does, is it is a place to maintain particular genes where you've synthesized that. So you collect them, right? That's what they do in these libraries. So you can take large sequences or cut into smaller pieces, pieces placed in vectors, the vectors introduced in cells, cells do what cells do, and they divide, and you all of a sudden have a huge library containing those genes because you're just letting the cells do what cells do best, and that's divide, okay? So that's pretty cool. So DNA sequencing. Okay, we can do this now where we can actually read the DNA code, determine the actual base pair sequencing. We are pleased. Fred Sanger, okay, this was his second Nobel Prize, in 1980, and we can actually read DNA. So pretty awesome. Okay, it has been very much refined. Okay, if you can imagine reading bands on gels constantly, and now we do it with very, very thin fiber optic gels that run through computers, which is pretty awesome. So there's a good old Fred. Okay, so this is how it has shifted over the course of time as far as cost per megabase of DNA sequence. That's why I can have our students do stuff and I can get it sequenced, because the price has come down so much just in the lab from 01 to 2011. I mean, look at that change. It's amazing how much the cost of sequencing has gone down, and that's because of the technology. So DNA gets replicated in the presence of dideoxynucleotides. Okay, so this is DDNTs. When it's incorporated into a strand of DNA, it terminates replication. The results process are read either manually, you can do it old school like with a ruler, okay, or automatically, which runs it through. So old school, they would literally get these bands, okay, and each band would represent a different nucleotide, your A's, your T's, your G's, your C's, and you just go from top to bottom, which one comes first, or this, okay. So the technique that they use now takes it, again, you get that those deoxyribonucleotides that are going to attach on. They fluorescently tag each one, okay, which is crazy, and you get this distinctive marking system running through a fiber optic tube of gel that works in the same, it's the same principle as electrophoresis, basically. Runs that laser through, detects it as it moves through the strands, and then you get your nucleotides and you get these waves, and you can read your order. So a little bit easier, nice and quicker than it was to do it the old school method when you were looking at stuff that was going on. Sometimes hard to tell which one came first, which one came second, and how it worked. So yeah, I can imagine my eyes going cross-eyed trying to do that. So that's how it is. Another form of reading is called a microarray. Visualize simultaneous gene expression for multiple genes. So if I want to know if something's being expressed on many genes at the same time, I can use a microarray. And so that's what we use there. So it creates this pattern that's there. And each well has a particular sequence complementary to that mRNA and how it fluoresces, and that's how we can know. And that pattern that gets analyzed, and that's an expression, analysis that we use. So here's the technique. You isolate mRNA from the tissue sample, okay? You make copied DNA by reverse transcription. So if I have the mRNA, I know what the DNA is, right? Apply that cDNA to a microarray of different genes in each spot, okay? So it's going to hybridize with any of the complementary DNA that's on the microarray, and you rinse it off and you scan it for fluorescence, okay? And each spot, each where it fluoresces would represent a gene that's being expressed in that tissue, okay? So here is just a fun, if we kind of zoom in on this here. So if you look here at this microarray, it's got 2,400 human genes on it. So each yellow spot would be a functioning gene in that particular tissue. So that's pretty stinking cool. So here, again, take a minute, and this gives you guys a visual sense of how microarrays work. Thus ends part 1. So welcome to part 2. In this part, we're going to look at the applications of all of these tools and techniques that we talked about in part 1. In this, to kind of find out what are ways really that we can use these. What good are they? So these are just some, some examples of applications of these. So let's jump right in. First one, genetic engineering, kind of a biggie, right? So what is genetic engineering? It's combining genetic information from 2 or more organisms to create what we call a transgenic, okay, changing, right, genes, organism to express new traits. This is going to involve restriction enzymes to chop and to resplice. It's going to include vectors. We have accomplished this through multitudes. Seems to be my word of the day. Through many domains of life. We can use it in medicine, agriculture, industry, research. It's, pretty awesome. So when you take a gene from one organism that is not found in another and you make it work in that other organism, you have genetic engineering. Okay, so for example, here is one way to do that. We take bacteria, we take the plasmid, we insert the gene into the plasmid, we put the plasmid back into the recombinant bacteria. So now we have it called recombinant DNA. Okay, so this Stanley Cohen and Herb Boyer, kind of pioneered this, back in the early 70s. And so you get your recombinant DNA bacteria. That's going to then grow in a culture. You're going to get boatloads of it. At Genentech, they have, double-story tanks of bacteria that are producing particular product. So it's pretty awesome. So because we know of the universalness of ribosomes and protein production, we can put it in there and we can do various things with it when we get that protein and actually harvest that protein. So you can work on the gene, you can work on the protein, you can do all kinds of things with it. So some examples, you might do genetically modified organisms, look for pest resistance, right? You could alter bacteria to clean up oil spills because they can metabolize that oil. proteins that can go in and dissolve blood clots to help keep our pipes clean, right? And things for like human growth hormone. There's also insulin. I mean, it goes on and on and on and on. All right, so the idea of making recombinant DNA, that's what it does. So here, check that off, another video showcasing the idea of recombinant DNA technique. So please feel free to go back and watch if you feel like you need to see this as well. I'm going to leave this totally in your own pause. So the details of it, recombinant screening. How are you going to know if what you did worked? Ways we do that, okay? So the frequency of getting those organisms to uptake those genes, things like that, it's pretty low. So you want to make sure that what you're doing is the most efficient and that you're really only using those. So in comes this idea of reporter genes that are going to tell you that you have taken up that gene. So examples of this are ampicillin resistance that I mentioned earlier, using green fluorescent protein to find out. Luciferase is another one. So green fluorescent protein, P-glow. Okay, so if the organism has taken up the gene, you should be able to put it under UV light and see it fluoresce, see the gene fluoresce bright green. Okay, so that's an example. The same thing with the luciferase. Okay, so here's your P-glow expression. This is a snapshot back from the P-glow lab. You've got your DNA, and this is all under UV light. You have the gene, bacteria that hadn't taken up the gene, that you didn't expose the plasmid to, growing in ampicillin, because ampicillin killed them all. Then you have adding the plasmid. You get it growing in ampicillin, but if you don't put the arabinose in to turn the gene on, then you get the actual, green fluorescent. So the fact that you have the ampicillin resistant identifies only the bacteria that took up the plasmid, whether that particular gene was turned on or off. So if you're going to go from here, you do this as a screening mechanism first, then you grab just those bacteria to make your subsequent generations from there, and that increases the efficiency of what you're doing. Here's as a reporter gene, using GFP is like huge. It's so important. So you can use various fluorescent proteins showcasing different gene expressions. In this case, we're looking at different ones within gene expression of fruit fly development. Okay, and the discovery of GFP and its subsequent applications won the Nobel Prize in 2008. So you can see how many, whether it's the medicine prize, whether it's the chemistry prize, whatever they may be, much of the Nobel Prizes can be tied in and associated with biotechnology. The advancements of biotechnology. And that's over, the last 40 years, really over the last 40 years with this sort of genetic biotechnology. Biotechnology as a idea and as a mechanism have been used for years and years and years and years and years. The idea of showcasing and using other organisms for the benefit, your benefit for mankind. That's been around for a long time. So another application is this idea of complementary DNA. I mentioned this earlier, cDNA, DNA copies of RNA molecules using reverse transcriptase. This is how a retrovirus works. It has an enzyme called reverse transcriptase, so something like HIV is a retrovirus, Ebola, and basically this enzyme takes the messenger RNA and goes backwards, creating a DNA strand. So it will increase your stability, because if I go from the already processed mRNA and create just the gene, I don't have to deal with all of the things that had been snipped out, okay, when dealing with eukaryotes. Because remember, bacteria, they will just, there's no processing. Okay, I transcribe my messenger RNA, that goes right to the ribosome, I make protein product. So if I do that, I can get rid of all of the stuff that normally gets processed and cut out. Now for eukaryotic engineering, it's a little bit more difficult because you've got the nucleus. There's a lot more different strategies that are involved in this, and we just like being complex. So if you, it's a funky thing, right, from Biorad, it's a gene gun. Okay, you shoot micro pellets micro, tiny, coated with DNA into the nucleus so that you can basically inject the gene that you want into the actual nucleus of the cell, which is like neat, right? Okay, so if I go in here, there's lots of different strategies. Agrobacterium is a good one because it has this naturally inserts the plasmid during its life cycle right into it. So you can use this kind of as a vector to take it and move it right into like plants. So a lot of times the plant, gene engineering can be used using this agrobacterium that infects the plant. So that makes that kind of nice as well. So just some basic ways. So again, eukaryotic is a little more complicated. So gene therapy, this is another application of all of our knowledge. Engineering cells for a multicellular organism to be able, to fix a problem. It's a little bit more tricky. You get some mixed results. So you take your clone gene, insert it into a virus. Okay, since we know what viruses do, they like to inject DNA into cells. We use that as sort of our transport mechanism to get it in there. Let it go in there. Let it infect the cell, okay, carrying the normal one into the bone marrow cell of the patient, and then you can inject the engineered cells into the patient, okay, and hopefully get it out. So this could be one way, to do that. So you use it in a retrovirus, it's going to do its thing, it'll, go back in. So which is kind of nice. It'll, excuse me, it'll be your transport for those genes. So that's one way for gene therapy, basically using these viruses to bring as little nano bots to bring in the gene that you want into the cells. And of course, if you inject it into the bone marrow, that's where you got stem cells, and then they're going to divide, hopefully dividing it now with that new gene. And then there's really complex engineering pieces, multi-gene traits becoming more common. So you use one organism with multiple gene things to create multiple products is what you would do there. So that makes it a little bit more so. You've got a goat that produces the blood clotting protein in its milk, so you can do that and put that in, and it's producing it so that you can get the animal being sort of the pharmacological factory for you. So getting that gene in, then you do that, collect it in the milk. Okay, you can also have golden rice, an engineered variety to have beta carotene in it in issues of nutrition depletion. So the beta carotene is already in the rice. That's why it's a little bit orange. And it's an important precursor and things like that for vitamin A. So when you have that deficiency, if I already put it in the rice, testing. Okay, so there's a brief moment in time when I thought about becoming a genetic counselor. So to be that person, that go-between that's going to help families understand potential issues with genetic complications, becoming a hugely more important field now that we have more and more abilities to test for things, as a result of genes. So using the tools, you look for the presence or the absence of particular genes. Latest story comes out of the fact that you're looking at potential people that would have contributing DNA from 3 parents, okay, instead of 2, particularly for moms that carry any kind of genetic abnormality. Okay, so lots of pros and cons to this. Again, that's one of those, yeah, we can, but should we, kind of things, and we'll talk about that. So restriction enzymes you use for the DNA digest, PCR for amplification, gel electrophoresis for analyzing, and then you have somebody who can help you understand what all that means. Okay, so typically when you do this, you analyze what's called a restriction fragment length polymorphism, okay, also known as R-flips, and this helps us look at these particular situations where we can learn about lots of stuff. So we are usually analyzing that. Yeah, we can use these R-flips and for looking for particular genes, okay? We can also look at helping to determine identity, areas of the genome that have many repeating sequences, so your short tandem repeats, these are STRs. You can rule out, that's the one thing you have to really understand about this. Anytime you do any kind of identification using DNA technology currently, you are never 100% identifying. What you're doing is you are ruling out other options. So you're basically, it's a way to whittle down the possibility. You take out what it can't be. and you're left with only, with lots of factors that can say whether or not it's really likely you've identified the right thing. So you can compare enough, you can identify, it can be confirmed or it can be ruled out, because, and mostly what you're doing is ruling out that kind of thing. So a lot of this is being used right now for inmates using DNA evidence to help determine whether or not the right person was put away, because at the time you basically didn't have the technology sometimes before, when they were found guilty. And now as the technology kind of improves, we can actually use that material and find out. And a lot of inmates have actually been released as a result of using DNA technology. We can do it for health purposes. We can look for genetic markers associated with various traits. We can look for things like sickled cell, normal alleles. We can find out if that's what's being carried. So you have the single-length polymorphism. That's just an expression for the cut that's made with restriction enzymes, if I didn't mention that earlier. That can be due to one SNP. It can lead to a loss of a restriction site, which then can be visualized on the gel. So if you're missing it, it won't cut there, and you'll be left with not 3 bands like it shows up in Part B, but with 2. And with luck, down the road, your whole genome you'll be able to have sequenced and carry on with a little card. And there's lots of questions with that too, as to who gets access to that information once it comes in. And then, of course, research. So, really the only limitation that we have right now is the barriers that the research has provided with us. So someday, maybe some of you guys will be involved in this, and if we don't have people questioning and wondering and being curious and going out there, we don't find stuff out. So this part becomes huge, as far as our ability to discover and learn. And then there's cloning. So I'm going to spend a little bit of time on cloning. We'll talk about it. It's one of those things where, again, that should we, could we, what do we do with it? Cloning, you're creating an exact copy of a genetic sequence. And when you hear people talk about cloning, of course, they're thinking about, here I am, and whoop, we create a whole nother me. Probably not exactly what the actual applications of cloning aren't just that, because you got to remember, to create me, a second me, a few things have to happen. Yeah, I mean, there's a few things that would have to happen in order to have that second me. You would have to get the embryo. There would have to be a mom to house my little embryo self for 9 months as I developed, a mom to give birth to me, and then you would have to raise me from a baby. So yes, while that baby would be my identical twin, is it me? I don't know. But it is a clone. That's what identical twins are. They're clones of one another. In reality, a lot of the cloning pieces right now are for pieces. Can you clone organs? Can you clone things like that? And we have been able to do it with a lot of different organisms at that point. In all domains of life, we have achieved a form of cloning. The first, very most famous mammal that was cloned is Dolly. Back in in the mid-late 90s, Dolly was cloned and over in Scotland. You can actually visit her. She did die in 2004, but she's now on display in Glasgow. I think it's Glasgow. I'll have to look that up. And so there's that form. So that's reproductive cloning, where you actually take the process to create a whole brand new organism. We do this with plant cuttings all the time. You take a plant cutting, you grow a new plant from it. That's a form of cloning. There's somatic cell nuclear transfer. Okay, so somatic cell nuclear transfer, and how we got Dolly started this way. The technique: you take the mammary cell. That's kind of why she was called Dolly on the side, like Dolly Parton. So I'll let you think on that one for a few minutes. The mammary cells were taken. You culture them. You get a bunch, right? You take out the nucleus out of her egg cell, out of the donor's egg cell, right? It wasn't from the same. She was not, the donor and the mom, okay, weren't in the donor, they weren't the same, okay? So you got the cell donor. Then you have the person who's going to donate the egg cell, or the sheep. You suck all the nucleus, the nuclear contents out of the egg. You take the information from the cultured cells and you put it into that, fuse those cells together, okay, and then you get a single cell with the nuclear contents from the donor. Okay, next part is let it divide, grow it in a culture, and you get an early embryo. As soon as you have that early embryo, you implant it into a surrogate mother, so the donor wasn't the mom. That would, a horse has done that, where the horse gave birth to its clone. That's weird. And you let it grow, and then you get a baby lamb. And that baby lamb Dolly was genetically identical to the donor who had given the mammary cell. Bah. So first successfully cloned mammal. And yeah, but here's the catch. It took forever. It took a long time. It took over 250 tries to get Dolly. So it's not very efficient or effective. And let me just say that many mammals have since been. Here is CC. CC is the first cloned kitty. She was cloned down at Texas A&M, and so she was the first cloned cat. They've cloned lots of mammals, but it's just not efficient. And then there's therapeutic cloning, and this is to be used for medical purposes using pluripotent stem cells from an early embryo. This can give rise to any, and we can culture cells for to heal. Maybe the idea of culturing stem, neurons and things for spinal cord issues that don't replicate in us. Can we put in new ones? All kinds of things. Get an organ so that if your organ fails, you have your own organ and you don't have to worry about rejecting it. So there's all kinds of things that happen with therapeutic cloning. And our last part 3, is going to deal with kind of that moral and ethical piece. So there's just some things that I want you guys to kind of consider when you're talking about the ethics. At what point, kind of how much do we have a right to know, right? There's the consent. When you're eating something, do you have the right to know whether it came from a genetically modified organism, genetically modified grown things? This is why there's lots of issues in the news with like Monsanto and a lot of other companies. Are they telling us what we need to? There's legislation to require require that companies place on labels that they're genetically modified. Okay, how you get your DNA sample. Should there be consent behind that? How much do other folks have the right to know about us? I mentioned if we have our own genetic code, who gets that info? So that's just something to kind of think about as we go through. The Assimilar Conference, 75, this was a big conference. Genetically engineered organisms that had started to be established. This was a big deal. There were many issues going on. Should we, could we, can we? People were very nervous about the possibility of what genetic engineering can do. Who owns it? Can a gene be owned? Can an organism be owned? If I clone a gene or synthesize a gene, who has right to that? Okay, so things like the ability to patent genes is kind of a big deal as well. When the BRCA1 protein, this is for breast cancer gene, was discovered, does the discoverer have stuff for that? What Monsanto does. Again, who owns this when you're dealing with something that's life? And then kind of the last big thing is, what does life mean? Okay. Does life mean something different, whether you're talking about a bacteria, whether you're talking about a mouse, whether you're talking about us, a tree? How do we define it? Should there be boundaries in terms of what we can do to other living things? This is where we get the embryonic stem cell debate for humans. Huge debate, because in order to do stem cell studies, you have to take that embryo and pull it apart, which obviously means it will never become a person. So is that okay? What do we do with that information? So are there boundaries? A fast aging knockout mouse. Okay, so 4 or 5 months, 8 months later, we've aged it. Is that okay to do to the mouse? The genetically engineered salmon. Look in the background. I mean, look at the size difference right there. Do we know what these genetic engineering organisms do for us when we think we're doing good things? Down the road, is it really okay for that? There's lots of things on that. Okay, a cloned rhesus monkey. We've put into these organisms the GFP protein, so these monkeys will glow under UV light. Okay, and this was used for lab study. Okay, early embryo, removal for cells to do stem cell research. This is, taking those apart. So where's life start? Where's it begin? Lots of questions to be answered. So again, that yes, we can, and can we do this? Should we do this piece is a big deal. Wow, so that was big. So with that being said, here are the things you want to make sure you can do with this, the tools and techniques, be able to explain the tools, what they are, their inputs, their process, their uses, their, what are the applications of these, how do we use them in sort of real world possibilities, and the ethics involved with them. So, just should we? Just because we can. All right, so with that, enjoy, and we'll catch you guys later.